Hello, my name is Ethan Wolfe, and today I will be discussing the work I've been doing to characterize effector-metaeffector pairs in Legionella pneumophila. I've been working with Joseph Burke, Dr. Stephanie Shames, and Dr. Janani Ravi. Bacterial effector proteins are virulence factors that are critical for parasitism in eukaryotic hosts. These effector proteins are pretty commonly used across a wide variety of pathogens. However, these meta-effector proteins, which are effectors that regulate the activity of cognate effectors, were recently discovered in Legionella pneumophila, which is the causative agent of Legionnaire's disease. These meta-effector proteins have not yet been identified in any other species. Meta-effectors are important for Legionella pneumophila's virulence. Strains that lack the meta-effector MESI are very attenuated for replication within host cells. This project focuses on 7 effector-metaeffector, or EM, pairs that contribute to virulence. The short-term goal was to characterize the sequence structural features and molecular evolution of the metaeffectors and study the co-evolution of the EM pairs. In the long term, we hope to develop a comprehensive feature repository that we can use to identify metaeffector and EM pair candidates. Let's talk about our approach for building this EM feature repository. So, we started with 7 EM pairs that are virulence factors, and we use this web app called EvolCompGen to search for homologs of these proteins. From NCBI RefSeq database, we got around 130 Legionella genomes, including around 100 Legionella pneumophila, and we scanned these Legionella genomes for our homologs of the effectors and metaeffectors. We then constructed an EM presence-absence matrix for all Legionella pneumophila genomes. We added our co-occurrence patterns, or we're going to add the co-occurrence patterns from this matrix into our feature repository, and also to be added to our feature repository are Wolfe domain architecture and phylogeny characterizations. Let's take a look at our results. So figure 2 here is our EM presence-absence matrix, and along the columns you can see the 7 effectors and or EM pairs that we have been studying. And each of the rows is a different Legionella pneumophila genome. So there's 102 rows and 102 Legionella pneumophila genomes. So each cell tells you whether the given effector or metaeffector is present or absent in the given genome. Then, based on the patterns in this matrix, if you look at figure 3 here, we were able to get a few basic statistics, or counts if you will, for each EM pair. So the first row tells the number of Legionella pneumophila genomes where both the effector and the metaeffector of that pair, so for example SIDI and MESI, are present. So if you look at SIDI and MESI, there are 97 Legionella pneumophila genomes in which both the effector and the metaeffector, so both SIDI and MESI, are present. The second row of this table is the number of genomes in which the effector is present alone without its metaeffector. 3 is the number of genomes where the metaeffector is present alone, and 4 is the number of genomes where neither the effector nor the metaeffector is present. Next, we also looked for homologs of our effectors and metaeffectors outside of Legionella pneumophila. So we wanted to see if there were any other Legionella genomes that had these effectors and metaeffectors, and we did in fact find a few homologs, most prominently SIDI and MESI. About 30% of our non-Legionella pneumophila Legionella genomes had some homolog of SIDI and some homolog of MESI. Then if we look at figure 5, this is a Wolfe visualization of the domain architecture of select metaeffectors. Unfortunately, the figure is a little bit tiny here, but we can see the annotation of a few different metaeffectors pulled from several different databases, and we can see leg A11 has some ankyrin repeats, and we also found that the meta-effector LubX is predicted to have a transmembrane region. So let's get to a few of the discussion points. So one interesting observation is that some of the effectors never appear without their cognate meta-effector in any of the Legionella pneumophila genomes. So these include SID-I, RAV-J, SID-H, and STVB. And we can hypothesize that these may lead to cytotoxicity within Legionella pneumophila. And then, across Legionella pneumophila, the effector RAV-J and its cognate meta-effector LegL1 are either both present or both absent. So within the bigenome matrix, you can see a very clear-cut pattern, and it's actually quite interesting to observe. Also, the EM pairs RAV-J LegL1 and SID-H LubX appear at a lower frequency across Legionella pneumophila, and do not appear in any non-Legionella pneumophila Legionella genomes. So we hypothesize that these EM pairs are not as critical for virulence as the other EM pairs are. Let's talk about future work. So one thing we can do to study the co-evolution of the EM pairs is to compare the phylogenetic trees of the effectors and their cognate meta-effectors. And this is there's something called the mirror tree approach we can use to doing this. We also want to apply our methods to other bacterial toxin-antitoxin pairs, and we want to continue to compile our results into our feature repository to work towards identifying novel meta-effectors and EM pair candidates. So I'd like to thank you for listening to my presentation, and I'd also like to thank the MSU Professorial Assistantship Program for placing me in the JROBI lab.